Good afternoon. Uh, my name is Someru Bhattacharya and I am in the Department of Pediatrics at the University of Rochester. And I would start with thanking the organizers for giving us an opportunity to present our results on single cell transcriptomic profiling of newborn human lungs. So this is, if I am presenting, this is not work done only by me. All the contributors primarily from University of Rochester and Cincinnati Children's Medical Center and our collaborators at Harvard UCLA Medical Center all have contributed to this work because, as we know, team means it's everyone works together to get more. So I'm gonna give you a little bit of background and description of how we ran the project, how the methods involved, followed by the cell type assessment in the newborn human lung, and then characterization of these newborn human lung cells using mouse lung cells. So the human lung is a complex organ. It comprises of over 40 different cell types. So far, mainly animal models have been used to define the mechanisms of diversity in the developing lung, and the understanding of the relation among the cells in the human lungs is lacking because there is not enough information available. More recently, the molecular analysis, including transcriptomics or proteomics, from sorted dissociated cells of human, newborn, and pediatric lungs, have indicated good quality and retention of in vivo phenotype. These have been performed as part of the Developing Lung Molecular Atlas program. And also in terms of single cell sequencing, we have recently worked with our collaborators in Harvard UCLA to create a singular cellular level landscape of fetal human lungs. So as we know, with the rapid advancement in the single cell transcriptomic technology, it enables the identification of cell type specific from transcriptomes of different organs. And using that, keeping that in mind, we decided to use the newborn human lung and apply the single cell transcriptomics to characterize the cellular lineages of a newborn human lung. So in order to do that, the human lung lobes from two one-day-old subjects were protease dissociated into single cells using methods that have been previously described. Then these unfractionated dissociated cells were rapidly thawed and without any testing were used. Each was used for two captures of single-cell RNA-seq on a chromium 10x platform. In one of the captures, the dead cells were removed by magnetic selection. And then subsequently, the libraries were prepared and sequenced on Illumina HiSeq. And then the data was analyzed using CURAT, including application of canonical correlation analysis for integration. And then we further used the cluster markers through top fun annotation to get the cell type associations. So give you an idea of what is involved in here. So we have two newborn human lung donors who donated the lungs to the human for research, and one of them was a full term and the other was a preterm. Both were just one day collected, one day post birth, and using this single cell sequencing, 2 runs on each, we combined the 4 runs together into one and then filtered for UMIs less than 500 and mitochondrial counts greater than 12.5% to keep it as close to a single cell, and with these criteria used to leave out potential doublets. So when we combine the four runs from the two donors, and we got like 16 clusters, and here I'm showing the distribution of the cells from individual donors, and look, you can see there is almost similar distribution in each cluster from both donors, where there are dark the black color for donor 1 and gray is donor 2. And then we again used SURAT and decided to look at individual run level distribution, and then again we could clearly see that even within the donors there is pretty good distribution across the four runs in each of the individual clusters. So that's pretty reassuring. And then we looked at identification of major cell types using the known markers, and as you can see that pretty using these epithelial cell markers, we have a few clusters that are epithelial cells, then majority of these were mesenchymal cells, some endothelial cells, and surprisingly for a fairly young lung, a newborn, we still get a good size of immune cells. And we further looked at the cellular subtypes or within each of the major cell types, and here is a distribution of cell landscape. We could find some 81 epithelial cells, 82 epithelial cells. Immune cells could be classified into B cells, T cells, and a lot of macrophages. And there are many subtypes of the mesenchymal cells, including myofibroblast and matrix fibroblast. And it will give you an idea of the distribution of the number of cells, and they are fairly equivalent. Maybe in some cases there is over enrichment or over representation of like 82 cells in the one of the runs, not in the other, but barring that we have good distribution across the run. So to summarize this part of the result, we did transcriptional profiling of the newborn human lungs, and we could see clusters of multiple populations of endothelial cells, fibroblasts, pericytes, and smooth muscle cells. We did not get very many epithelial cells. However, whatever we saw had a good representation of the epithelial markers. And as I said before, for lungs that young, we still saw multiple clusters of immune cells. And then these marker genes for the individual populations were used to analyze the regulatory pathways, and then in an attempt to distinguish similar populations of the cells. Once we know the uh, cellular landscape of the newborn human lung, the idea is to, well, even though all of our runs were from 1 day old lungs, we know that the cells of in the cell lineages in lung, what we have found in the rodents, that there are differences even within cell types in terms of age, and this has been seen in the aging lung. And since the lung development process is pretty conserved across human and mouse, but there are differences such as the relative length of the stage, and one of the most critical differences, the timing of birth. In human lungs, the birth occurs during the alveolar stage, while in mouse it takes place in saccular stage. So keeping that in mind, we decided to look at the single cell RNA-seq data from newborn or 1 day old mouse. And our collaborators in Cincinnati had published this paper last year where they characterized the cellular landscape of 1 day old mouse lung. And using this data and additional transcriptomic data from postnatal day 3, 7, and 10 generated by at Cincinnati, we integrated all four time points, and here is a t-SNE plot where you could see the integrated almost 33,000 cells from the mouse, one newborn all the way to like a mature mouse from 1 day, 3 day, 7 day, and 10 days old. These are the 30 clusters, and you could see a mixed distribution of cells from each time point in each of these clusters. And similar to human cells, we looked at the known cell markers in the mouse lung cells, and as we could, using the same, we identified them into major cell types. And in difference from human cells, we could see a lot more epithelial cell clusters in the mouse, but even in the mouse, we see quite a few immune cells and mesenchymal cells. Just to keep in mind that these are in fact from newborn all the way to, we can consider, equivalent to grown-up humans or teenager humans. So we would expect to see almost all cell types in here. We further looked at the cellular subtypes within each cluster, and we could find all the different cells that could be expected, including the subtypes of epithelial cells. We could see multiple clusters of matrix fibroblasts and immune cells altogether. So this is a rich data set from what we got from the mouse. And then what we did was we integrated the mouse with the human cells. And one thing, as we had mentioned before, that the birth in mouse takes place slightly earlier than human comparison. Like in human, the birth takes place in alveolar stage at 39 weeks, while in mouse it takes place at saccular stage, which is the embryonic 18 days. So in this data set, we combined the two cells and gene expression, the transcriptomic profile, using 14,502 genes that were matched across species through Biomart. And then we integrated our 5,500 human cells with the 32,000 or 33,000 approximate mouse cells. So the human cells comprise 15% of the whole object. We use this canonical correlation analysis to combine the two objects, and then even in the integrated data set, we ran the same steps of group combining, generating them into clusters, and then using markers to identify their cellular types. Just to give you an idea, we had the newborn human lungs. We did single cell sequencing and combined them into one object, annotated individual cell types. We had mouse lungs from 1, 3, 7, and 10-day old, and we did a single cell sequencing, combined the mouse data, and then we further integrated the human and the mouse data sets together. That gave us 33 and 5,000 or 38,000 cells. And this is how the integrated data looks like. The gray are the mouse cells and the black ones are the human cells. And as I pointed earlier, that the human cells in general are the 15% of that However, we did see cellular differences where the like mesenchymal cells were much more, while the epithelial cells were very few. And then we further looked at separating them, and here is the outline. So you can see almost in every cluster there is some human cells. And we looked at the known major cell type markers. We could identify them into the major cell types: endothelial cells, epithelial cells, mesenchymal cells, and immune cells. And using the known markers, here's a heat map of the known markers. The yellow color indicates high expression, and the maroon or purple is low expression. And using the markers that are highly expressed, we did find the further cellular subtypes within each of those clusters. And here is a distribution for cellular types and subtypes across all four runs of the human subjects and all four runs of the mouse or mouse. So we could generate a mouse lung universe and then integrated both the human and the mouse lung, which is not only integration across species but across two platforms. And then we analyze this integrated human mouse lung cells to reveal multiple clusters of major cell types and as well as the subtypes. And the human cell proportion varied by cell types, with mesenchymal being close to 25%, however, and the epithelial cells being less than 5%. So next, what we did was we attempted to estimate the maturation state of the human cells. So we know both the human lungs donors were 1 day old. However, as evidence or as presented previously by others, even in aging lung, that the maturity aging say varies by cell type. So we are hypothesizing that the individual cellular development also varies by cellular cell types. And in order to do that, we decided to look at the maturation state, but estimate that in terms of the postnatal days of mouse lung cells, because we are going to look at if the mouse data is varying across time. So we are going to look at the human cells in terms of their proximity to the different ages of the mouse cells. And we did this process separately for individual cell types, not all of the 33,000 cells together. We used principal component analysis to reduce for dimensionality reduction. We identified one principal component that is associated in the mouse cells with age, and using that principal component, we looked for the nearest mouse cells from every human cell, and based on the proportion of the mouse cells and weighted by the total number of cells for that type, we of that age, we calculated the ages of each of the human cells. Just to give you an idea how it went, here is a schematic of integrated cells. We split them out into individual cell types. We looked at we ran PCA and the PC associated with the age for every human cells. In this plane, we calculated the ages, and using this equation, we identified the age range for every cell type. If you are confused, don't worry, because I do have a schematic for you. So let's start with, say for example, we are going to look at the endothelial cells. So pull them out and plot them here. Just the endothelial cells, that are gray ones are the mouse, the dark ones are the human cells, and then a PCA. And here I'm showing a PC1 and PC3 plot. As expected, in PC1, the biggest separation factor is the human as a species. So the red ones are the human, and these green and brown are the mouse cells. That's expected. Then we look for the ages, the principal component that is associated with age. And for example, in the endothelial cells, you see the principal component 3, which is shown right here, uh, is the one associated with age, and it had an absolute, the PC loadings when correlated with age had an absolute correlation of 46%. Use now on this age of principal component on the plane of PC of age PC3, we measured the distance of every human cell with every mouse cell. So for example, again here the yellow one is the ones that are closer, and the purple ones are, I'm sorry, the yellow ones are farther away because the higher value, and the purples are closer. And then using this matrix, we'll look for the closest mouse cells for every human cell. And we looked at 100 closest mouse cells, and using this formula where you're taking the average of the frequency of each of mouse cells from each age, weight, age is factored in their age, as well as using a weight. Weight is not the mouse weight in terms, but weight is actually the proportion of the that mouse cells of that age in that particular cell type. And using that, we estimated the ages of the endothelial cells, for example, here, which range somewhere from 7 to 9 days. And now, when we looked at all the cell populations individually, you could see here the endothelial cells mostly were in the 7 to 9 days, but there's some down below. Macrophages pretty close, between 5 to 8 days. B cells 4 to 6 days. CD4s in the same range, 5 days. My fibroblasts. However, in matrix fibroblast, you can see two groups. One that is ranging right here between 2 to 4 days, and the other is in this. And when you're using a frequency plot, it is even better visible. While all of them are within the range of say 4 to 8 or 9 days, the matrix fibroblast here are two different groups. And if you recall that a newborn human lung is closer to a 6 to 8 day old mouse lung in terms of physiology and pathology. So this cellular, this distribution of ages is quite expected. Everything is between 6 to 9 days. However, the surprising part is we could see two different groups of matrix fibroblasts. And then we looked at the genes that are differentially expressed between the two groups, and we found 219 genes that are differentially expressed. And out of these, 187 were overexpressed in the younger ones. And when we looked at those, they could see the pathways associated with smooth muscle contraction, extracellular matrix, collagen, oxidative stress, and all those were being affected in these younger fibroblasts. We also looked at a validation, and here you could see we looked at the markers for early younger matrix fibroblast and in general mesenchymal cells, as well as the markers for other cellular types where these markers for the early matrix fibroblast exist. And here you can see the ones that are marked in pink arrows are the ones that indicate the presence of the immature matrix fibroblast and expression by HES1, and any of the mesenchymal markers like either POL6, A3, or TCF21. While the yellow arrows here indicate in general matrix in general mesenchymal cell markers. So to summarize this part, we could estimate the maturity state of the human cells based on their proximity to the mouse cells. And most of the newborn human lung was developmentally similar to 6 to 8 day old mouse lung. However, there were differences by cell types, and where most of the matrix fibroblasts appeared to be relatively younger. So to summarize the whole project, we have successfully performed the single-cell RNA-seq from newborn human cells. In the newborn human cells, we could see mesenchymal and endothelial cells. We could see relatively few epithelial cells and some immune cells, considering in that younger lung. And we integrated the human and mouse cells together, and using the mouse cells we estimated the maturity state of human cells. What we saw as maturation state differs by cell types, and the matrix fibroblasts have most of the matrix fibroblasts were much younger compared to all the other cells. I would like to thank our collaborators both at the University of Rochester and other lung map centers. And here is an outline for the whole project, just for reference, and I would be happy to answer any questions. Thank you for your time today.